In this video, we are gonna see about polyclonal antibodies by Dr.G. Gayathri. Polyclonal antibodies (PABs) are antibodies that are secreted by different B cell lineages within the body, whereas monoclonal antibodies come from a single cell lineage. They are a collection of immunoglobulin molecules that react against a specific antigen, each identifying a different epitope. Characteristics of polyclonal and monoclonal antibodies. Monoclonal antibodies. Expensive production, long production time. Large quantities of specific antibodies recognize a single epitope on an antigen. Production is continuous and uniform once the hybridoma is made. Polyclonal antibodies. Inexpensive production, rapid production. Large quantities of non-specific antibodies. Recognize multiple epitopes on an antigen. Different batches vary in composition. Lab animals are usually injected at least twice with antigen when being used to produce antiserum. The second injection will activate memory cells that make class IgG antibodies against the antigen. The memory cells also undergo affinity maturation, resulting in a pool of antibodies with higher average affinity. Affinity maturation occurs because of mutations in the immunoglobulin gene variable regions, resulting in B cells with slightly altered antigen binding sites. On re-exposure to the antigen, those B cells capable of producing antibody with higher affinity antigen binding sites will be stimulated to proliferate and produce more antibody than their lower affinity peers. An adjuvant, which is a chemical that provokes a generalized activation of the immune system that stimulates greater antibody production, is often mixed with the antigen prior to injection. Clinical uses of polyclonal antisera. Polyclonal antisera are used in many clinical tests that are designed to determine whether a patient is producing antibodies in response to a particular pathogen. While these tests are certainly powerful diagnostic tools, they have their limitations because they are an indirect means of determining whether a particular pathogen is present. For this reason, antibody-based tests are often used only as screening tests. If the results are positive, other confirmatory tests are used to make sure that the results were not false positive. In addition to their role in diagnosis, polyclonal antisera can activate complement, detect the presence of bacteria in clinical and food industry settings, and perform a wide array of precipitation reactions that can detect and quantify serum proteins, viruses, or other antigens.